Hello, today we're talking about experiments, and as all experiments are, these are based off of the shoulders of the scientists that come before them, so there's a lot of background information that's important to getting to this point, but we're just going to talk about what they did in these specific experiments. So Nirenberg and Matthaei, they wanted to determine what specific RNA sequence coded for a specific amino acid. At this point, we didn't know how many amino acids were in a sequence or what the sequences could code for. So what they did was they created a cell-free system using E. coli bacteria and created synthetic RNA molecule. For this example, they used poly-U, which is just a long chain of U's, so that way they would know that whatever was created used U's only to create it. They filled 20 test tubes with different amino acids, where 19 of those amino acids are cold amino acids, as in they're just normal, and one is tagged with C14, that is hot, and each time they tagged a different one. So they would do a set of 20 where histine was the tagged one, and then they do another set of 20 where lysine was tagged to determine, rule them out until eventually they found out which one UUUU coded for. They would introduce UU to each tube, and then if there was a protein produced, they would test it to see if it was radioactive, and if it was radioactive, then we know that it used UUUU. So they found out that UU codes for phenylalanine, and then they found out AAA is lysine, CCC is proline, and GGG didn't do anything. They couldn't test anything else, because if they had done a UAA, for example, they wouldn't know if it was UAA or AUA or AUU, because they didn't know how many, or like what order they were going in, or how they were taking. They didn't know how to do the frames of a start codon and an end codon, so they just had to do all U's. And so Nirenberg teamed up with Leader to determine the remaining codon sequences and their corresponding amino acids, and they used very short artificial RNA sequences that were only 3 nucleotides long in a cell-free system. The shorter length fragments were long enough to allow ribosomes to bind with the type of tRNA molecule that is complementary to the one codon, and it was still able to be detectable. So they mixed together all the necessary parts of DNA, and then they labeled one type of amino acid at a time and put the mixture through a millipore filter. The filter allowed unbound tRNAs to pass through it, but did not allow the ribosomes with the bound triplet to pass through. So the sample was then tested for radioactivity. If there was radioactivity found in the sample that did not pass through the filter, the corresponding amino acid had been attached. So they found they were able to decode the remaining 61 codons. The reason they weren't able to do 64 was because there's 3 codons that code for stop, so that wouldn't create a protein and wouldn't bind to any ribosomes, so they weren't able to decode those, but process of elimination, you can kind of assume that that's what those do.